I am at the Center for High Technology Materials, which is on the south campus of UNM. And this talk presents an invention that was the result of a joint work with Hugh Smythe, who is at the University of Texas at Austin, Lisha Armijo, who now is Lisha Martin, since she got married, and she's currently at the Texas A&M University in Corpus Christi. She was at that time my PhD student, and Nihal Bandara, who was a postdoc with Hugh Smythe in Texas, and who is now at the University of Bristol in the UK. This particular invention is a spin-off of an NIH award that was primarily aiming at the drug delivery to the lungs of patients suffering from cystic fibrosis, where in particular Pseudomonas aeruginosa is a bacteria that invades the lungs and is very difficult to treat because it hides behind the biofilm. So we would be using nanoparticles with the antibiotic to deliver the drugs. The invention was filed initially in 2015, and we just recently in June received a note from the US Patent Office that the application has been allowed. So by September, we expect the patent to be issued. So in the interest of time, I will skip most of the slides that are here. In case if somebody wants more details, I'll be happy to share those slides with anybody who requests. And if you didn't note my contact information, Greg certainly can provide it easily if anybody interested. These are my co-inventors, Hugh Smythe, Nihal Bandara, and Lisha Martin. The biofilms are discussed in this slide. Again, in the interest of time, I will not go through all those details, but the main issue is that if you want to deliver drugs, the biofilms represent an obstacle that the antibiotics cannot penetrate. That's why we were using nanoparticles, in particular super paramagnetic nanoparticles that could penetrate the biofilm. And in the process of that research, we have discovered that the nanoparticles, even without the drug, actually have bactericidal effect. And this was the basis of this invention. And then we started thinking, what could it be used for aside from the drug delivery? And the application we focused on, kind of very close to the previous presentation, is the gingivitis that, if not treated, turns into periodontitis. And again, I will skip most of that because this is just the background, but it's a very widespread disease that in the US, almost 50% of the population suffers that. The UNM Dental Hygiene Department told me that 80% of their patients have problems with gum disease, gingivitis, or even more advanced periodontitis. So while gingivitis itself is reversible and it's not that damaging to the teeth, when it is not treated early enough, it could lead to periodontitis. And that one is a very severe disease that can eventually lead to the loss of teeth and also has side effects such as increased risks of heart attack and stroke. And it's irreversible as opposed to gingivitis. So what is our solution? Our solution is to use those super paramagnetic iron oxide nanoparticles that we can synthesize here at CHTM. And they can be mixed either with mouthwash or toothpaste or maybe even with the gel that you just heard about, and expose the areas that otherwise are not easily accessible to the effects of the iron oxide nanoparticles. The iron oxide itself is safe and biocompatible, so this is a very good material. And while we have focused on gingivitis prevention and periodontitis, there are many other possible conditions which I will show later on in a slide that these nanoparticles could be used. So here is a brief summary how the biofilm or dental plaque is formed, and it involves 3 layers of colonizers, where the first colonizer, shown at the bottom here, binds to the teeth surface by using the proteins from saliva. And these colonizers are from the Streptococcus family. Then there is a second layer, which is this magenta color Fusobacterium nucleatum, that is an intermediate layer on which the third most damaging colonizers, such as Porphyromonas gingivalis, for example, can attach to. And that's what leads to actually disease. So in our experiments, we are attacking primarily this second colonizer because there is a large number of first colonizer, a large number of third colonizer, tertiary colonizers, but Fusobacterium nucleatum seems to be a universal necessary second layer without which the third colonizers cannot establish themselves. So we have conducted a number of experiments. This shows the cultures of Streptococcus gordoni, and Fusobacterium nucleatum on its own doesn't want to grow that well, but when it's on top of surface of Streptococcus gordoni, then you have a very successful culture development. And then we conducted a bunch of experiments using Listerine as a mouthwash, comparing that to the effects of the nanoparticles, and of course having control samples. So this slide shows the first set of experiments in which the effect of the deionized water itself was examined compared to the Listerine. And as you can see, Listerine is more effective, significantly more effective than DI water. So about 50% of the bacterial colonies were killed by DI water itself, while Listerine kills more, like 99%. And here is a comparison of the Listerine and the nanoparticles. So we exposed the cultures to 30 and 60 seconds of nanoparticles with a concentration of 8 micrograms per milliliter. And as you can see, the effects of mouthwash after 60 seconds are pretty much the same as the effects of the nanoparticles after 30 seconds. One of the problems with nanoparticles in these experiments was that they were in liquid suspension and tend to separate themselves from the liquid by agglomeration. So this is not necessarily the ultimate result when it comes to capability of nanoparticles to kill the bacteria. Now, the current status of the work is that we are in final stages of preparing for experiments that will determine what are the safe levels of the nanoparticles to healthy gingival cell cultures. And once we know those, then we will compare the bactericidal effects of the nanoparticles at those safe levels with the efficacy of toothpaste, which in my opinion is much easier to have the uniformly dispersed nanoparticles than a liquid such as mouthwash. Now, in terms of investment or return on investment, the global oral care market is huge. By 2025, it is expected to reach about $60 billion, and it has a steady growth associated with increased awareness of the dangers of poor oral hygiene. There is a bunch of companies shown here as the key industrial players that we are all familiar with, because these are the toothpaste and mouthwash producers. The other potential applications are listed on this slide. There's plenty of those, wound treatment and other applications where the bacteria can be killed using iron oxide, which is much more gentle to other cells than silver, for example. So thank you very much. And if I have any questions, I'd be happy to.
Thank you, Marek. Do we have any questions out there? Please feel free to type in the chat. One question I have, there's, the improvement over Listerine versus the particles. It is better, but have you got any feedback from the market of whether people would be willing to pay a premium for an improvement, like slight improvement over the standard, which is Listerine right now?
Listerine is not effective in preventing gingivitis or periodontitis in itself. So the problem is that it does not stay long enough. Well, the nanoparticles will, and therefore the action of the nanoparticles on bacteria could be prolonged. Another advantage of paramagnetic nanoparticles is that if needed, they can be removed using the magnetic field.
I don't think we have any other questions. Thank you again, Marek.
And they're also cheap.
Oh yeah, that's good.